Thank you. Yeah, my name is Willem van Hoorn, if you want to hear the Dutch version. I work for a company, Extensia, which used to be 2 words, but then so many pharma companies had laid off people, so that everyone thought I was Extensia. No, that means from knowledge, yeah, Latin, bit pretentious, but that's basically what we do. Data mining, AI, to discover drugs faster. Yeah, so where do drugs come from? There's a little star there, I don't know if you can read, but it says legal drugs here. Yeah? So this is all above board. Yeah? So you may have heard 1 in 10,000. Yeah? 1 in 10,000 compounds that pharma companies try becomes a drug, and you get that funnel picture. Yeah? You pour in 10,000 compounds that you allegedly have on the shelf, and you do some different levels of filtering, and the blue is the clinical trials, yeah, where you actually give it to people, and then out pops one drug. Is this true? Not really. It's only partially already. I have a clicker. Yeah, so this bit is true. Yeah, from here onwards, it's really a funnel. Yeah, you have drug candidates, and then you give it to actual patients, and you see if it works. Yeah, so that's where compounds drop off. But the top bit is bullshit. Sorry. We are not entirely true. Yeah? So it's not that pharma companies just have to buy 10,000 compounds or make 10,000 compounds and just have to fish out the drug. That's not how it works. Yeah? So how does it work? So like a lot of engineering things, it is prototyping. Yeah? You design something, you try something, you see if it works, and then is it better, yeah, or not? Right? So you learn from that experience, and then you know the next prototype. Yeah? So that is how James Dyson built his vacuum cleaners. Apparently, allegedly, he started with cardboard boxes, and now you have the shiny bagless vacuum cleaners. Yeah? But there were hundreds of prototypes in between. And drug discovery is very much the same. Yeah? You start with what we call a hit. Yeah, so you have a compound with some of the desired activity. What does that mean? A compound is just a chemical. And when I talk about activity, that means it does something in the body. Let me keep it as vague as that. But it doesn't only have to be active. Yeah? If you want to make a pill, it has to get into the body and not, let's say, come out the other end unchanged. Yeah? So there are a lot of constraints. And usually when you start here, you just have a compound that does a little bit of what you want, but not enough, and of all the other things it has to do, it doesn't. You have to optimize it. Yeah, and traditionally, medicinal chemists, those are the chemists who design the drugs, traditionally, come up with ideas like, oh, I can improve this, let's make that. Yeah, so they start with one or a few, expand to lots of ideas, but then you have to make them, yeah? Synthesis, chemical synthesis in the lab, and then you have to do some biology to measure the activity. That is slow and expensive, so you only have all the ideas, you can only do a few. Do I have a drug candidate? Yes. Then I go into the funnel, but usually no. Yeah, you update your knowledge, and you have the next ideas. So usually you go through this cycle at least 20 times, and you make each round 10, 100 of compounds. Yeah? And this can easily take a month or a couple of months. Yeah? So that's roughly how drug discovery works, how it has worked for a long time, and also how we still do it. Yeah? This design, make, test is still valid. It's just that we do it differently. So if you look at historical productivity, so how much effort does it take to get a drug to market? Yeah? So historically, companies made between 2,500 to 5,000 compounds in order to get that one candidate. Synthesis of each of them is about 5,000. Yeah? So that's quite an outlay. And it takes about 4.5 years. Yeah? So that's really, really expensive and time-consuming. And that has been like that for a long time. Yeah? And if you look around in the world, yeah, algorithms these days, yeah, decide whether you get your credit card and how much interest you pay on it, filter what ads you see, yeah, trade for you on the stock market if you have a pension, if you're lucky enough to have one. Depending on the app, introduce you to potential long or short-term partners, etc., etc., etc. Yeah, so beat humans in chess, Go, Strategio, do your homework, ChatGPT. Yeah? And drug discovery hasn't changed. Well, it has. Yeah? So companies like ours and our competitors and also Big Pharma, we're all trying to use these kind of technologies in our drug discovery. Yeah, so this is a marketing slide a little bit. Yeah, so instead of 2,500 to 5,000 compounds in 4.5 years, we can do it in 250 in 1.5 years. Yeah, I mean, it's not. We've done it a couple of times, enough to say confidently that we can. It's not a guarantee for the future. Yeah, it's like investment. But, so how did we do this? And it's all about strategy and tactics. Yeah? So yes, it says strategy without tactics is the slowest route to victory. So if you know where you need to go, but you bumble around, you will eventually get there, but it's not very efficient. And so tactics without strategy is the noise before defeat. Yeah, if you go really fast, but you have no idea where you're going, yeah, you don't get there. That's roughly what this says. And so we use AI, but we don't use it to replace the human. Yeah? Humans are very much part of the design, make, test cycle. Humans are good at strategy and judgments. Yeah? So this is the kind of things, which is a bit nerdy maybe, but if you work in a drug discovery project, compounds come in series. Yeah? You make little modifications, but there is a theme to all those compounds you make. And you say, oh, all of them have the same issue, they inhibit hERG. You don't need to know what it is, but you need to know it can kill you. Yeah? So that's definitely something you don't want. Or you can say, oh, we have a model, a machine learning model, but it doesn't really perform. Yeah? So that's the kind of human judgment. And then the AI, the computer, can do the tactics. Yeah? We use generative methods. You say, okay, design me some derivatives of this chemical series where the predicted hERG activity is lower. Yeah? That is what an AI can do for you. Or we apply active learning, and I come back to that. I say, okay, design me some compounds, and if I make and test them, then I know I get a much better model. Yeah? So optimize your learning. Yeah, so I will show you two examples. One about machine learning, and we're going to build a model, so pay attention. And one about a few about active learning. Yeah, so machine learning. Yeah, so what do we actually do? Yeah, so we predict activity. Yeah, how well it binds to a protein target. So maybe how good the effect is of your compound, your chemical structure. Given the chemical structure, yeah, and this is how chemists look at molecules. Yeah, they draw these kind of. This is high school chemistry. Yeah, you fake maybe you still have nightmares, but yeah, ring systems, and these are atoms with bonds. Doesn't really matter. But we're going to build a machine learning model in our heads. Yeah, so because I took here 5 random compounds, and these are 5 compounds that are not random. They all bind to a protein called MMP12. Yeah, and if you can help, well, maybe not on this monitor. And if you would stand here in front of me, you could maybe spot the difference. But there's a clear difference between the two. Yeah, for me it's obvious, but I'm biased. Yeah, but basically, these look very much like each other. Yeah, this bit of the molecule here in the middle is the same for all of them, while here can be anything. Yeah? And basically, a machine learning model picks up on that. It said, hey, hang on, there is a pattern here. Yeah, so machine learning, to some extent, is statistics on steroids. It just says, hey, I've seen this 5 times, 5 times it was active. Never seen it in an inactive compound. So that this bit must be very much associated with activity. Yeah, so here's the quiz. Given all of these structures, do you think this is a MMP binder or not? It's not a trick question. Maybe it is a trick question if you sit further away, but yeah, it is one. Yeah, like that's that same bit here in common. Yeah. Yo, we built our model successfully. Yeah, that is what it can be as simple as that. Yeah. So it is a bit more than that, but that's how it starts. And now the other thing, active learning. Yeah. So and that's all about making those design, make, test cycles efficient, yeah, because if you can imagine, you make multiple compounds per cycle, but if you make 10 of them and they are nearly identical of each other, it's like investing in one stock. Yeah? If the stock does well, you do well. If the stock does not well, you go down. And it's the same with here. If you make the right one, you're in. But if you make the wrong one, you're 10 times the wrong one. Yeah? So it makes much, for a learning point, makes much more sense to spread your risk. Yeah? Not all eggs in one basket. And then we call that exploitation and exploration. What do I mean with that? Yeah, so this is the time of year where two of my hobbies collide. One of them is mountain biking, and the other one is mushroom hunting. Yeah, it's autumn. So the task is tasty mushrooms in my backpack. Yeah, that's and we have resource constraints. Yeah, there's only so much I can put in the backpack, and there's also, given my age, so much distance I can ride. Yeah, so how to find the best mushrooms given my resource constraints? Yeah, that's an optimization problem. Yeah, so I can do exploit, yeah, which means I use my prior knowledge and I just use that to pick mushrooms. Yeah, so these parasol mushrooms, they're roughly this time of year, it's like September, October. And I roughly know where they grow, yeah, because I have my mountain bike routes and I see them every year. So I roughly know where they are. So I could sort of say, okay, let's go there, pick them. But you can also see they're fairly big. Yeah, this is my hand. And this is a small one. So I can pick maybe 3 or 4 of them and my backpack is full. Yeah, but they're nice, but they're not great. Yeah, so I could just settle for nice, or I could go a bit further. Yeah, explore areas where I haven't been, and maybe find much better ones. And these, trust me, are much, much better. These are seps, the king of mushrooms. Really, really nice. But you can also find things you were not looking for. These are true pictures I took. This I took last year. It was a humongous number of fly agarics, and someone tried to make a heart, or picked a lot of them, tried to pick a heart and love in mushrooms. Yeah, it's a bit macabre because it looks like bones, but it's just made out of fly agarics. And I am innocent. I only took the picture. And this I found in the middle of the woods. So I think someone was, whatever, scraped off the bike dots and sniffed them. I don't know what happened there, but yeah. Things I didn't expect. I didn't pick either of these, yeah, and I would strongly recommend not doing that. So. Yeah, so that's exploration, exploitation, and how in a more mathematical basis do you do that? And there's a technique called design of experiments. Yeah, and I try to illustrate this with this picture. This is a stained glass window. It's actually in a Cambridge college. And there's something special with this window. Yeah, so if you look at the red colors, there is one red square in each row, but there's also one red square in each column. And the same is true for all, I think it's 7 colors, did I say that? Yeah, 7 colors. So these squares are not randomly arranged, there's a pattern in there. And that's called a Latin square. I don't know why it's called a Latin square, but it is called a Latin square. And you can use that to sample. Yeah, this is actually from Wikipedia, but you can imagine there are many ways you can arrange 7 different colors on a grid by 7 by 7 grid. Yeah, you cannot really read this, but this says 7, and this is a number which is trillion, the trillions. Yeah, so people calculate if you have only one color in a 1 by 1 grid, but one option. In the 2 by 2, 2. But yet this is like exponential growth. Yeah, and since COVID, we know everything about exponential growth. This number goes so big that with 12, we don't even know what the number is. Yeah, so you cannot do this exhaustively. Yeah, but this you can use to maximize your return of information. And how does that work? Yeah, so now we do a little bit of chemistry. This is a textbook reaction. Yeah, you go. I'm pointing my finger. That looks better. Oh, no, wrong way. Come on. Yeah, here we go. This says R1, and this says R2. And basically, this is an acid. This is an amine, and these two react. You get a chemical bond. You see R1 chemical bond R2. So basically, it's Lego. Yeah. red bricks and blue bricks. Yeah? And you have different bricks, and they're marked R1 and R2. So if you have 5 acids and 5 bases, you can make 25 products. You have 5 by 5. Yeah, so we call that, this is my design space, 25. That's all the ideas I have. Yeah? That's the limited medicinal chemist can come up with 25 ideas. But given the size of the lab I have and the budget I have, I can only make 5. Yeah? Which one should I make? 5 out of 25. Yeah? So you can use your Latin hypercube sample. Yeah? You can just say, well, make here are my 5 R1s, here are my 5 R2s. Well, make the diagonal. Yeah? Make each different R1, each different R2. I use at least once. I've seen what they do. Or I can do this one. Or 160,000 different ones. Yeah? Which one to choose? Yeah, so, but now we bring a little bit of exploitation to break the tie, because maybe I already have a machine learning model. It's not very good, it's a crude model, but it's better than just random. Yeah, so that's basically the only criteria I have at this stage. It needs to be better than random. So if I color these squares by the model score, yeah, this is a fictional example, say red is good, blue is bad. Yeah, so model says no, model says yes, but I cannot really trust my model. Yeah, if this was a perfect model, I would just make these 5, yeah, the red bit. But I cannot really trust the model, but I cannot really ignore it either. Yeah, so what do? The Latin hypercube. I pick the best compound, best predicted compound. That's that one. And then I'd say, okay, I've now seen this R1, I've now seen this R2. I'm not going to use those again. Yeah, and you can fill in how you do that. You get the diagonal. Yeah? With this particular model, you get the diagonal. But basically, I use that model to break the ties. And that is a very efficient way of sampling these kind of matrices. And I can show you how that works. Okay, this is an example for literature. People published a matrix of 50 by 50 compounds. Yeah? These are real made compounds and measured data. You can also see reality collides with theory. There are white spots in there. Yeah? Not all syntheses work, and not all biological experiments work. So there are blanks in there. And I've colored it by inactive to active. Yeah? Blue to red is inactive to active. Let's do some searching. And you can do, let's start with the 10 worst compounds which are in there. Yeah? They're here. And let's do some exploration first, and then my model gets trained, and then do some exploitation. Yeah, so here we go. This is iteration 0. Here are my 10 worst compounds. Here is where I need to be. Here is where the good compounds are. But if I build a model on these 10 compounds, they're all bad. Yeah, so what does the model think? It thinks the whole matrix is bad. Yeah, because it has only seen 10. They're all bad, so the prediction for the rest is also, wow, they must be rubbish. Yeah? Now we're going to sample this. So I pick one compound, look up what the activity is, update my model, use that model to recolor the matrix, and then pick the next one and the next one. Well, you will see how quickly a model learns. Yeah, you see, it has only picked 13 compounds now. That's already figured out. This is where I need to be. But because if I use the Latin hypercube sampling, I force it to sample here as well. It knows already it needs to be there until I've done 50, which is now. So now it has done 50 out of 2,500. And the model sort of already has spotted, like here is where I need to be. And now I just release the constraints and say, okay, now pick just the best compound. So we go to exploitation mode. And that becomes a bit boring because it just fills in the columns. Basically, every compound it picks now is an active, is a good one. Yeah, so in the end, I have now done 250 almost. Yeah, we're done 250 compounds. So I've done 10% of the matrix. Found all the actives. Yeah? So basically I went from the 10 worst to the 10 best and many more in 10% of the effort. Yeah? So that's how we do drug discovery. I can show you a real life example. Yeah, this is how people sample their chemical space. People like rows and columns. It's also easier for the synthesis. But this is a real life example. The colored ones were the ones that were made, and the little circles were the ones that they proposed to make. Yeah? So they wanted to make more of these. That's not very efficient. We told them that's not very efficient. And they said, okay, show me you can do better. This is from their past. Yes? Company data, similar as before, yeah? Partially filled matrix, and said, okay, let's turn that into a game. We start here, and then this human versus computer, yeah? Human versus algorithm. What would you make next? And that, you know that game. And Bill, remember Tuesday? Okay, so there's an intermezzo here, but here's a battle cruise with 5. Yeah, so our answer in the pub quiz was right. Yeah? So we know. But
we were right.
Yeah, we were right. And in this version of the game, we were right. Anyway, let's do it. Okay, yeah, we did that. Our algorithm versus 10 experienced medicinal chemists. And this is basically what happens. Yeah, there was a kind of score. If you pick good compounds, you got rewarded. If you pick bad ones, you got penalized. If you do this random, you get this, yeah, because there's slightly more good compounds in there than bad ones. If you cheat, yeah, if you know which ones to pick, you get this. So this is the theoretically maximum score. And this is the compounds in the order they were picked. Well, you could see that a very simple algorithm at the time beat 9 out of 10 chemists. Yeah, they were very happy to be our customers at that time when we did this. It's also, now we have that data set. A couple of years later, I rewrote the algorithm, and now it's basically as good as chemist number 2. It's kind of funny, we had a meeting, and everyone claimed to be chemist number 2. We all got a t-shirt, I am chemist number 2. But yeah. So basically, we're now to the conclusion. So yeah, in the modern world, yeah, chemistry and biology are still hard and slow and expensive compared to running an algorithm at Amazon, AWS, or wherever you want to run it. Yeah, so compared to wet chemistry, wet biology, yeah, machine learning is a commodity. It's fast and inexpensive. Yeah, and drug discovery is not a big data problem, it's actually a small data problem. Yeah? So how can I be as frugal as I can be, make as few compounds that I need to make and not more in as fast as I can, yeah? Because the company down the road is also chasing this disease, and the first one to get a drug on market gets a huge reward. Yeah, so essentially, okay, yeah, we are expert-led, that's the humans, AI-driven, and we try to automate part of the drug discovery as much as we can. We're also now building robots who actually do the synthesis. But you cannot only do this with a computer science background. Yeah, you need to know the problem you're working on. Yeah, so some of our competitors, brilliant computer scientists, but yeah, sometimes shockingly naive when it comes to chemistry or drug design or medicinal chemistry. Yeah, so I think we have a competitive advantage there. Yeah, you have to think carefully where to leave the human in the loop. Yeah, we as humans are not redundant, and that's, yeah, I can show you. ChatGPT wrote me the abstract, but okay, let's not go there. Yeah, really. Okay, well that was it. Thank you. Any questions?
Any questions?
So with written AI responses, we get things like hallucinations, where
Yeah.
it will make something up.
Yeah.
Do you see the same with chemical?
Yeah.
You go and create an element and you'll go, what?
Yeah, so the question is also about hallucinations of large language models like ChatGPT. Yes and no. So we care a lot about what we call domain of applicability. Yeah, so what I said earlier, it is basically statistics on steroids. Yeah, so if you have trained it on certain chemical features, and then you give it something completely different, yeah, then the model doesn't know what to do. But usually you notice because the score goes to 0, which means like, oh no. Or there is a separate applicability score that says, well, yeah, I think it's brilliant, but I have no idea what I'm doing. So there is some baked-in warning. Plus, yeah, we make those compounds. Yeah, we make and test them, and especially in the beginning of a project, when your model is trained on tiny data sets, we know it's dodgy, but it doesn't have to be perfect. Yeah, so it only has to be better than random. Yeah, then that already is a competitive advantage. Another question.
That's really interesting. Apologies if I might have misunderstood. Is it the case that in this sort of process, would you use whether open access or provide access academic research to train the model that then becomes a proprietary model?
It's a mix. There are a lot of machine learning. Basically, companies have to disclose data. Yeah, if I want to bring a drug to market, I have to patent my invention. And if I patent my invention, I will say, well, I made this compound, and here I show it works. Yeah, so you have to publish structures and activities. So patents are public. The disadvantage is they are PDFs, yeah, and computers don't really read PDF files. They want, yeah. Okay, we get into a long discussion of how do you represent a molecule in a computer, but not as an image in general. Yeah. So you can buy that patent information, but there is also a database called Campbell, funded by the Wellcome Fund, as it happens. And they pay people in India who also go through all the literature, and they say, hey, there is a table here, and there is a molecule in here, and in the text it says it has a certain value for activity. Let's put that in a database. And that is publicly available. Yeah, you can go home and download it. It fits on a laptop. But the data we generate in-house is ours. Yeah? And that is not only, I mean, we're not unique. Every pharma company, the crown jewels, to a certain extent, is the compounds they've made and the things, the activities they've measured. And so that is, if I want to, for instance, when I showed you those molecular structures, I had to be really, really careful that none of them were proprietary. Because if I show here a proprietary structure, that means I cannot patent it anymore. Yeah? Because this is public, especially since you upload it. But that counts as a disclosure. Yeah? So for patent law means I cannot patent it anymore. Suppose I've shown you here the molecule that cures whatever. Yeah? It's useless, yeah? Because if you cannot patent it, anyone can make it, which basically means no one will make it, because it is still quite expensive to make it, but you cannot really charge a lot, because everyone can make it. Yeah? So there's the kind of bind we're in that patents are what keeps us profitable. Well, we haven't a single drug on the market, so we're not profitable. But what keeps pharma companies profitable, but they only last 15 or 20, I never know, years. So for a while you have a monopoly, and then suddenly everyone can make your compound. Yeah. So when I worked for Pfizer, it was an extreme case. When I worked there, Pfizer sold Lipitor against high cholesterol, 10 billion a year. That's like an insane amount of money. And then the patent expires, and then the year after they did 5 billion, yeah, and then the year after it's 100 million, which sounds like a lot, but compared to 10 billion is nothing. Yeah, you go from boom to bust to boom to bust. Sorry, I'm rambling here. Yeah, so the models are proprietary, but the technique how to build those models is, yeah, we use open source software for a large part for that, and we're not that different from others. So the competitive advantage is usually not so much in the machine learning itself, more how you combine them, how you execute. Yeah, so if you can do your chemistry quicker than someone else, then your design cycles are shorter, you reach to market quicker. Yeah, if you were the first to patent, there's a huge I call it non-linear reward. Yeah, it's like World Cup football. No one remembers who came second. Yeah, and that with drugs is the same. If you're the second on the market, yeah, the market's already gone by that time. Yeah? So there's a huge drive to be the first on the market. Yeah. Well, that drives innovation. Michelle.
I'll make it an easy one. Hi. I don't know how many in this audience have ever watched 2-minute papers? No? Okay, so there's this guy, what's his name? I can't remember his name. We watched it at home, it's kind of about entertainment, but he is really into generative AI.
Yeah.
And so he is showing, if you tune into his YouTube channel or whatever, he is showing that you can, describe in vision language, generate, generate a photo-like image of a cat playing fiddle on the couch on its over the moon, and he'll come up with multiple, and this is neural networks, obviously, it's generative AI. How does that compare to machine learning? Tell me what the difference.
Okay, so the question boils down to the difference between deep learning and AI, and also generative AI, roughly. So that's an interesting one. Some people think AI is the same as deep learning. Yeah, so deep learning is the thing that drives the ChatGPT, yeah, which is a large language model, which basically, in the end, is just, remember predictive texting on your phone, yeah, on steroids. Yeah? Instead of predicting the next letter, it predicts whatever the next paragraph. Yes, but in the end, it is the same. It is statistics predicting what humans actually want to read. What is the most logical next word here? So I think deep learning is just a different way of doing machine learning. Yeah, so the one that I showed you, which beat the chemists in 2015, was basically a naive Bayesian classifier, which is roughly 200 years old, the algorithm. Yeah, the implementation is slightly newer, but it is, and I think all of them are useful. Yeah, one is really quick, and you can train it probably on the computer which is in my watch, yeah, as simple as that. And the other one, you need half a data center, and the underlying model of ChatGPT costs about 10 million, I think it was 10 million to train, just in GPU hours that you need. Yeah? Just the Amazon bill or the whatever Azure bill was about 10 million. So you and I probably don't have that the back of our couches hidden. Yeah? So that makes that not really accessible. And also running that model, yeah? Half the world is using it, and every time you run it, yeah, there is some GPU working for you. That's expensive stuff, yeah, that they offer it for free. That long term they can't. Yeah? Long term there has to be some monetizing. But yeah, those generative models are amazing, yeah, especially in images. But we use them as well to dream up molecules, but then it's very easy to come up with, let's say, molecule-like structures, yeah? Just like artists can draw molecules on a cover, and that's usually funny when a chemist look at it, like, that's not a molecule, yeah? The classic one is the right-handed or the left-handed helix, when people depict DNA, that often goes wrong. But there are also some chemical structures that just, you can draw them, and they may exist somewhere in interstellar space at whatever 0 Kelvin, but at room temperature they don't exist. Yeah, so and there are many more ways of making a wrong structure than a correct structure. Yeah, so there's a lot of training involved to have them produce valid structures. I think that is now more or less a solved issue, but do they do it better than simple, let's say, classic chemoinformatics methods? I'm not really convinced the new methods work better, but yeah, I'm in my 50s, so I'm a bit in that sense an old technology guy. But yeah, they are probably the future. Yeah, and some companies, they ditch, let's say, the methods I used. Yeah, so a lot of this is early stage of what we did. Yeah? My younger colleagues thinks I'm an old-fashioned person, but yeah, generative AI is really powerful, and I think we haven't seen what it can do in drug discovery or in society in general. Yeah, I mean, ChatGPT, it's the spam emailer's dream. You can generate hundreds of different emails at the click of a button. Yeah.
Yeah, and the first, we've been watching these 2-minute papers over time, so the publishing results in the first time, the cow jumped over the moon. Well, it didn't make much sense. It does suggest that over time, it's going to be self-improvement.
Yeah.
Yeah, so with machine learning, I guess one has to...
Well, you saw the design, make, test cycle.
Exactly.
Yeah, every cycle you update the model. Well, that's
but the neural networks versus,
and collectively, every time we pick out whatever the traffic lights and because we capture.
But do you think that, I guess,
we're training a model.
AI is that it's kind of got a bit of a black box, and you don't
Yeah, well,
know how...
yes, because those models contain literally billions of parameters. Yeah, so you cannot really trace, like, you can see at the end, this comes out. You know at the beginning this went in, but the number of transformations that happen in between are so, I mean, that will cost you your life, that will take you your life to figure that out. Yeah, that's...
And then where's the knowledge created?
Yeah. So that's a bit of a disadvantage. So how do you distinguish a proper prediction or a proper picture from hallucinating? Yeah, the model itself doesn't know. Yeah. The model doesn't know. The model is not a human. Yeah, let's not. It's an algorithm.
I got a question about the background. Because I don't know that, is there a finite number of possible molecules?
Oh, it's an interesting one. Yes, there are estimates for that in the sense that, so we live in the world of organic chemistry, which means we only use, let's say, 8 elements out of the, 100 in the periodic system. So that's already a limitation. That's carbon, nitrogen, oxygen, hydrogen, what is it? Chlorine, fluorine, I'm forgetting what, sulfur, that's roughly it. And then you could say, okay, most drugs have a maximum size. And you can sort of imagine why that is, because basically if they become too big, well, basically you cannot dissolve them anymore. And if you cannot dissolve something, you cannot absorb it in your body. Yeah, so there are some natural limits.
So there are natural boundaries.
Yeah, so if you take
all of possibilities of that.
Yeah, so if you take those boundaries into account, people have estimated you can roughly make 10 to the power 60 different molecules, which is more than atoms in the universe. Yeah, so
then I got a following up the question, because you mentioned that then if the model gets a completely new piece of data, you won't be able to work on it, and therefore distill it to something potentially useful for us.
Yeah.
How is that relating to that number? Will we, with that sort of learning, be able to create...
Well, so there are more constraints, and the biggest one is you actually need to be able to make it. Yeah, so you can...
Okay.
You cannot... usually drugs have a ring system, yeah? The pictures I showed you, you saw those rings, and you can merge them, so you have merged rings. And you can just write down what are all the possible rings of 12 members which have whatever 3 nitrogens in there. You can enumerate them. And then you look in literature, how many have been made? 0.1%. Yeah? So the thing is, if you want to make the others, you can calculate with some quantum chemistry, is the thing actually stable? You think, yeah, it's stable. Do I know how to make it? No. Yeah, so time is a big constraint for us. Yeah, so if you have to make something completely new and you don't know how to make it, then maybe you have to fund 5 PhD students who work whatever 4 years each on it, and then we have a new synthetic route. So long story short is to say that we tend to be conservative. We restrict ourselves to chemistry we know, which means roughly compounds that are not too far away from compounds that we have made in the past, so we know probably we can make them. Yeah? So
So then I get another question that. Yeah. Okay. Is that the case then? Within some time, we be able to explore potential all of the possibilities in terms of drug, and then from that point, we only be wondering how to actually make them.
Both, I would say. Yeah. So it is like a trade-off. You want things to be novel, and that means it's new enough that you can file a patent. Yeah, that it doesn't look something that is very close to what other people have done, but not so novel that you have no idea how to make it. Yeah. So you want to make a step forward, but not a very big step forward. Yeah. So that trade-off, like exploration, exploitation. Yeah. That's always, that basically I live in a world of trade-offs. I'm always doing this at work.